Sorry about that. Okay, word. Hi, everybody. This is our review session for the second half of this week. We're going to be talking about RNA interference and gene expression regulation with the trip operon. Okay, let's get this started. I know it's just you, Sev, but do you have any questions so far, like before I jump into any of this?
No.
Okay, word. I'm going to be trying to check out the chat every now and then. So if you want to like privately message me or Rachel or just put like a general question, I could try to be on top of that too.
Yeah, I can look at the chat too while you're talking, Sebastian.
Word. Okay, thank you.
Yeah.
Okay, so RNA interference, it's essentially this really neat way for the cell to it kind of destroy mRNA or any kind of RNA that it produced that it ultimately doesn't want. So let's say a cell needs this channel protein for whatever reason, and it starts transcribing a bunch of mRNA transcripts to get this channel protein all over the cell. And then the cell realizes it made too much mRNA and it doesn't actually need all of those mRNAs becoming proteins. So one mechanism that the cell has to correct this or to destroy all that excess messenger RNA is to produce these two kinds of RNAs that we call RNA interference. There's two kinds. There's two main kinds that we'll be talking about in this course. MI, which is microRNA, and SI, which is small interfering RNA. It's really important to know the differences between the two. I'll definitely, I'll of course be like going over the differences between them. They're not extremely different, but there are some differences that are worth noting. So with that being said, do you have any questions before I move on by any chance?
I haven't listened to these lectures yet, so.
Oh, okay, awesome. So we're going in blind. Okay, so I'll try to be like as detailed as possible. And I'm sure maybe whoever watches this later, if you want to like pause the video or slow down or anything, feel free to do that. Okay, so real quickly, this is, okay, sorry. Like I mentioned, there's two main pathways. There's the microRNA and the siRNA. So I'm first going to be talking about the microRNA pathway. So the miRNA, this is what the cell does naturally, as opposed to the siRNA, small interfering RNA. That's something that you have to like put into the cell yourself. It's not something that occurs naturally in the cell. So that's one difference, but again, I'll cover the main differences later. MI, so microRNA, is something that the cell does on its own. So what happens is, okay, let's go back to that channel protein example. Let's say the cell made a bunch of mRNA for this channel protein that ultimately it doesn't want all that mRNA that it produced. So what the cell is going to do is it's going to transcribe straight from its own DNA. It's going to transcribe this really, really, really long piece of RNA called the pre-RNA. I know the diagram here says pre. This is actually a mistake on the diagram. This is the pre-miRNA. This is the pri. It's just a little typo on the diagram. But anyway, the cell's going to transcribe this really long single-stranded piece of RNA called the pri-miRNA. Okay. Drosha is this enzyme inside the nucleus, which cuts the pri-mRNA, and after it cuts it, it turns it into pre-miRNA. So that's why there was like a typo here. This is supposed to say pri. After Drosha cuts the pri-mRNA, it turns into pre-miRNA. Another way to know that you're talking about microRNA is whenever you talk about Drosha. So Drosha is an enzyme that cuts the pri-mRNA, but it only exists in the nucleus. It's never going to be outside of the nucleus. So if you ever have a question that says, or whatever, like a problem that's talking about Drosha and it keeps mentioning Drosha, it should be like a red flag that we're talking about miRNA, because Drosha does not involve small interfering. It does not involve siRNA. So that's another difference. But again, I've said this like 3 times already. I'll probably say it a 4th time. I'll cover like all the differences later. But whenever you see Drosha, that is something exclusive to the nucleus, which is exclusive to the miRNA pathway. So anyway, Drosha is going to cut the pri. Oh, and if at any point you have a question, you want me to slow down, feel free to stop me or interrupt me. It's totally fine. Okay, so Drosha is going to cut the pri miRNA and turn it into pre-miRNA. The pre-miRNA is going to be exported out of the cell, and it's going to be exposed into the cytosol. So now that you have this pre-miRNA, there's another enzyme in the cytosol, dicer, that cuts the pre-miRNA further to make it into these little miRNA duplexes. So essentially, the microRNA gets cut twice. It gets cut from pri-mRNA to pre-mRNA, and then the pre-mRNA gets cut further into just miRNA. So now that you have these little duplexes, now that you have these little mRNA duplexes, there is a protein that's going to find it called Argonaute. Argonaute is going to find these little mRNA duplexes, and it's going to hold on to them. Argonaute can only find the duplexes when they are double bound. So nobody really knows why, but for some reason, Argonaute cannot find miRNA when it's single-stranded. So it has to be in its duplex form. It has to be in its double strand form for Argonaute to find it. Again, the exact reason as to why is still kind of unknown, but for whatever reason, Argonaute only has an affinity for the mRNA duplexes when it's double stranded. So anyway, Argonaute is going to find one of these duplexes. And these duplexes is obviously composed of two parts, because this is double-stranded DNA, or sorry, this is double-stranded RNA. So it's composed of two parts. It's composed of one part that has exact complementation to the mRNA the mRNA transcript that you're trying to get rid of. So again, just to like refresh everything that I've talked about, the whole reason that the cell is doing this is because it's trying to destroy mRNA that it doesn't need. So that mRNA, of course, has its own nucleotide sequence. These mRNA duplexes, one strand has the exact sequence, and then the other strand has the complementary, so the opposite sequence. What Argonaute is going to do is it's going to unwind the double-stranded RNA, and it's going to do away. Actually, let me ask you, which strand? It's going to do away with one strand, but which strand do you think that Argonaute is going to get rid of? Do you think it's going to get rid of the strand that has complete complementation, called the sense strand, or do you think it's going to get rid of? Sorry, let me rephrase that question. Argonaute's going to get rid of like one strand, but is it going to get rid of the strand that has exact complementation, or the strand that is exactly the same sequence? What do you think?
The one that's the same sequence.
That's the one that it's going to get rid of.
That's what I would think, yeah.
Yes, that's exactly right. Because the miRNA has to be able to bind to the mRNA that it wants to get rid of. So if it keeps the strand that has the exact same sequence, it's not gonna bind to your target mRNA. So it's going to get rid of the strand that has exact sequence, and it's going to keep the strand that has complementary sequence. So that complex, that Argonaute with the mRNA, that's called RISC. It was RNA interference sequence complex or something like that. This RISC complex, is then going to find its target RNA, and it's either going to like sequester to a P-body so that the mRNA could get destroyed or recycled, or it's just going to like immediately chew up the mRNA. But that's essentially how miRNA destroys that target mRNA. Okay, so I know we've been talking like forever, kind of incoherent rambling, but do you have any questions about anything that I just said? I have steps. Actually, here are the steps of everything that I just said. But do you have any questions so far about micro mRNA or this pathway?
Yeah, my one question is about, so Argonaute, that because that's not pictured on the diagram, correct?
Yeah,
So
not illustrated here.
so Argonaute comes in, and it is what, I guess, so the miRNA duplexes are formed because Argonaute cannot recognize the single-stranded.
Yes, well, kind of. The mRNA, these little can you see my cursor by any chance?
Yes.
Okay, sweet. These mRNA duplexes are formed because the cell here transcribed like a really, really long piece of mRNA that's single-stranded, but single-stranded RNA is going to like fold onto each other because that's just how base pairing works. When it folds onto each other, that's how you get these little like hairpin loops. So that really long piece of RNA again is going to like fold on itself to form these hairpins. Drosha is going to cut that long piece of RNA to make it into like smaller pieces of hairpin loops. Dicer is going to continue cutting it so that you're just left with these double-stranded things, the double-stranded duplexes. This is what Argonaut is going to find.
And where does the complementary strand to what Dicer created, where does that come from?
It's somewhere here on the duplex. So this mRNA duplex, again, it's not illustrated like in the best way, but oh geez. Let's say like let's say the bottom strand is the strand that is exactly identical to your target mRNA. And let's say the top one that's illustrated with like this red line, let's say that top one is the complementary strand. So Argonaut's going to find this duplex. It's not exactly understood how it knows, but somehow it knows which one is the complementary one, which is the red one, and somehow it knows which one is the one that's identical. It's going to it has like a helicase function, which allows it to unbound it to separate the two strands, and it could just get rid of the one that has exact similarity to the mRNA. Does that answer your question by any chance?
Yeah, that makes sense. Thank you.
Sweet. Yeah, no problem. Okay, so that was mRNA. Oh, and something worth pointing out. This mRNA transcript, so once RISC is just the complex of Argonaut with the target miRNA. That's all that RISC is. It's just a complex. That target mRNA, the RISC is holding onto, doesn't have exact similarity to the mRNA that it's trying to get rid of. It has like a pretty good amount of similarity, but not 100% similarity. And that's actually intentional, which I'll talk about in a little bit. I'll talk about later. Okay, so this is mRNA, again, just to point out some of the differences or just some key different points as opposed to siRNA. The fact that it's made in the nucleus makes it different from siRNA. siRNA is not made in the nucleus. When drosha is involved, drosha only exists inside the nucleus. So again, that's evidence that this is miRNA, and the fact that it doesn't have 100% complementation to the target mRNA is another factor that this is miRNA. So everything I just said will probably make a little more sense when I start talking about siRNA. So they're very, very similar, except the only difference is that siRNA, small interfering RNA, it has to be injected into the cell. So again, this is not something that happens naturally. It's not something that happens naturally in our cells. It's something that we have to physically like inject into the cell. So what we do is we inject double-stranded RNA, or this is a small hairpin RNA. So it could be double-stranded like this, or it could be a hairpin. It doesn't really matter. The following steps are exactly the same as miRNA. So after you inject your siRNA, Dicer is again going to come, it's going to cut the double-stranded RNA or cut the small hairpin RNA. It's going to cut one of them or either of them. Doesn't really matter. Dicer is going to cut them. Argonaut is again going to find this and is going to make the RISC complex. It's going to get rid of the strand that has 100% similarity to the mRNA, and it's going to keep the strand that is complementary. So now that RISC has the strand that's completely complementary, it's going to find the target mRNA. So the mRNA that the cell wants to get rid of, it's going to attach itself and it's going to get rid of it. So it's essentially exactly the same as miRNA. The main difference is that it does not start in the nucleus. This is injected straight into the cell. It does not go into the nucleus. Nothing gets processed inside the nucleus. Everything that siRNA involves is the same as miRNA, except it gets exogenously injected. Another main difference is that siRNA has 100% complementation to your mRNA, to your target mRNA, whereas miRNA has a great deal of complementation, but not 100%. And again, this is just the explanation for everything that I talked about. So do you have any questions about small interfering RNA up until this point?
No.
Okay, sweet. Okay, so it's pretty common for Laura or Professor Francis to put some questions that ask you to recall the differences between the two. So again, I'm just going to cover the differences between mi and siRNA one more time. The differences is that the difference is that miRNA begins inside the nucleus, whereas siRNA begins in the cytoplasm. So that's one difference, which I think this sentence was trying to cover. mRNA has to be transcribed, processed, exported. So again, miRNA is something that naturally occurs in our cells, whereas siRNA has to be injected into the cell. So mi is a naturally occurring thing. Human miRNA is not 100% accurate, but siRNA is 100% accurate. And again, I'll talk a little bit about the different, or I might ask you what the differences are later. We'll see. siRNA is used. Oh, okay, yeah. siRNA is when it, like, let's say you're a researcher and you're trying to do like knockout experiments. So your cell that you're studying is transcribing a bunch of miRNA, or excuse me, it's transcribing a bunch of messenger RNA, but you have no idea what it's doing. A popular experiment to use is a knockout experiment where you knock out each individual miRNA, or excuse me, messenger RNA, and you observe like the phenotypic difference of what the cell is doing. So that's why siRNA is used in like therapies or experiments, things like that. So some of the similarities is that they both involve dicer, they both involve the RISC complex. Again, drosha is only specific to miRNA because drosha only exists in the nucleus. siRNA does not do anything with the nucleus, so anything that involves drosha is exclusive to miRNA. Both RNAs are unwound with one, yeah, and another similarity is that once you have that duplex, whether it be mi or siRNA, RISC is going to unbind them and is going to keep one as a guide, the one that has complementation, and is going to degrade the one that is exactly identical to your target mRNA. So any questions so far about the compare and contrast?
No.
Sweet. Okay, so do you have any ideas, maybe stirring in your head, as to why the miRNA is not 100% complementary? If you don't, it's okay, but like, do you have any ideas as to like maybe why that's the case?
It could be that we don't, I mean, with siRNA, we consciously, I guess, know exactly how much we need. But maybe with the, the human-made miRNA, we wouldn't, we don't know exactly how much RNA. I don't know if that really makes sense.
No, I got you. Actually, the first part that you said actually made a lot of sense because siRNA it's almost always used exclusively for like research purposes. It's almost only used for knockout experiments. So if you're a researcher and you're doing research on the cell and you want to know what all these mRNA things are, one experiment you could do is the knockout one, where you knock out a bunch of miRNAs, or excuse me, you knock out a bunch of messenger RNAs and you see how that affects the cell. So the reason that si, which is the one that researchers inject into the cell, the reason that siRNA has 100% complementation is because we're in control of what we want to knock out. We know exactly which mRNA we want to target and get rid of. So that's why siRNA has 100% complementation to that target mRNA. However, in cells, miRNA is not 100% complementary. And the reason for that is because it allows for the mRNA to not just target a very, very specific kind of message RNA. Because it's not 100% complementary, it could actually target a more broad range of messenger RNA. So let's say the cell produced like just a bunch of messenger RNA by accident, whatever. It's going to make miRNA, and because it's not 100% complementary, it's able to target a more broad range of messenger RNA. Whereas with siRNA, when you're doing an experiment that involves siRNA or a knockout experiment, you know exactly which mRNA you want to get rid of. So you're able to make 100% complementation because you know exactly what you want to get rid of. But if you're just a cell and you want to get rid of a bunch of messenger RNA that you made by accident, your mRNA is not going to be 100% complementary because you want to be able to knock out a greater range of messenger RNA as opposed to just one very specific kind. Does that make sense by any chance?
I don't totally understand. I would think that it would be very important to only knock out a very specific kind, because it doesn't want to, I mean, it doesn't want to degrade any mRNA that are needed, you know, to maintain cellular function
Right.
or
Yeah,
anything
that
like that.
yeah, that and that's definitely a good point. That's actually one of the cons to miRNA, that even though it allows the cell to target a greater deal of messenger RNA, the caveat to that is that it could accidentally knock out something that it might actually need. Of course, that doesn't happen. It doesn't happen too often. Otherwise, like if it was something that happened all the time, we would probably be like very messed up organisms. But considering the fact that we're like relatively okay, it has a pretty good mechanism to figure that thing out. But I totally understand like the apprehension. Is there anything you want to like add to this by any chance, Rachel? Anything that I just said about anything?
No, honestly, you're pretty spot on.
Okay, sweet. I'm just going off of like what Saeed said. He covered this section, and I just watched like his whole video, so he did a good job at like summarizing this. So, let's thank Saeed for that. Okay, what's the difference between mRNA, pre? I don't like this question. I'm just gonna talk about it. So, mRNA, you'd have to go back to this diagram. So, mRNA is just like the really, really long piece of RNA that folds in itself. Again, this diagram is supposed to say not pre-miRNA, but whatever. Pri-miRNA becomes pre-miRNA after Drosha cuts it. And then pre-miRNA becomes mature miRNA after Dicer cuts it. Okay, so that's the answer essentially to this question. All right, I think I am done with RNA interference. So do you have any questions about anything relating to RNA interference? I still recommend you watch Laura's video. Like, I'm sure she includes a lot more detail than than I did, so I still recommend you watch it. But
I honestly, I thought that this session was gonna, I haven't watched any of them yet, and I thought this was gonna be 2-1. I was like,
yeah,
I'm gonna watch these videos tomorrow.
Oh,
yeah,
gotcha.
no, you're
Two
fine.
ones actually already up on the YouTube.
Yeah.
Yeah.
Yeah, so I mean, I don't mind getting a
Like a sneak peek, I
preparatory.
guess, of what to expect.
Yeah.
All right, no worries.
But yeah, I usually would have watched the lectures before.
Yeah, that's fine. Wednesday, just so you know, for like next time, or for like whoever's watching, I guess like what Rachel and I have been doing is we kind of dedicate Wednesday's review to all of like the first half of the week, and then the Friday review to the second half. Does that sound about right, Rachel? We've only done like 4 of these,
Yeah.
like, so if you know if people want like, or they have like some feedback, like they want something to change, we could probably like work that out. This is such a small class that we can honestly like just do whatever.
Yeah, definitely.
Yeah, we're us and Laura, we're super flexible with how this course is going out. So, you know, if you want to talk about anything, just email us. We're
It's
fine.
easier to manage with only 15 students instead of 175.
God, yeah. All right.
My turn? Are you doing this part?
No, I'm not doing this part. This is all you.
This is all me. All right, sick. Okay. So, moving on to gene expression regulation. One of the ways that gene expression is regulated is through transcription factor mediated transcriptional regulation. So that's a mouthful. But basically, it's just using transcription factors to regulate the rate or activity of transcription. So why would we need transcription factors in the first place? So RNA polymerase cannot bind to DNA. So remember, RNA polymerase is what is transcribing the DNA. It can't bind to DNA and start transcription without transcription factors on the DNA. So transcription factors control RNA polymerase binding to promoters in genes. And there's certain types of transcription factors. There's general transcription factors and regulatory transcription factors. So general transcription factors are basically, it's in the name, they're general. I know you guys had on your problem set that question that a few of you were confused about because she hadn't gone over it yet, but sigma factor in prokaryotes is a transcription factor, a general transcription factor, and in eukaryotes, they're called basal transcription factors. So general transcription factors can be used at many different promoters. That's why they're called general. They're not promoter or gene specific. Whereas regulatory transcription factors are required to control transcription at specific genes or promoters. So that's the main difference between the two of them. The regulatory ones are gene specific. And when you hear the terms positive and negative, so basically positive regulators enhance transcription when they're bound to DNA, and negative transcription factors suppress transcription when they're bound to DNA. So the main takeaway that you should remember from this is that RNA polymerase plus a sigma factor in prokaryotes leads to the initiation of transcription in prokaryotes. And in eukaryotes, it would be RNA polymerase plus the base factor for general transcriptional regulation. If you're going to be specific, then it would be regulatory, but Laura likes to focus a lot on sigma factor, especially in like her questions on the exam. So actually, you can go to the next page, Sebastian, because the diagram's bigger. There you go. All right, so this diagram shows you have your RNA polymerase, this blue thing where Sebastian's cursor is. That yellow circle in the middle is the sigma factor, because we're just talking about sigma factor here, which is the general transcription factor in prokaryotes. And then you can see that little section of green on the DNA is the promoter, and then right where that orange starts is the start site. So basically what happens is the sigma factor binds to the promoter first, which allows RNA polymerase to come in and bind to the sigma factor. And then that allows transcription to begin. So then on the next section, the RNA synthesis begins. That's that little complex there. And then eventually the sigma factor will diffuse out because now the RNA polymerase doesn't need it anymore. And then it can rebind if it needs to do more transcription after. But the main thing about transcription regulation is that without the sigma factor or other transcription factors, RNA polymerase cannot bind to the DNA and cannot start transcription. So a little bit more on transcription factors. So they can bind in a sequence, specific manner. So I'll talk a little bit about this, I think, on the next slide, but so in prokaryotes, we're just going to continue with sigma factors. E. coli has 7 different ones, but sigma 70 is the one we really focus on in this class. They're used in the normal growth phase. They bind to about 800 promoters in E. coli. That's why they're general transcription factors, because they're not really that specific in terms of which genes they bind to. And different promoters have different affinity for sigma 70. That's how it's regulated in terms of whether or not it can, if sigma 70 can't bind, then transcription can't really occur because that RNA polymerase wouldn't be able to also bind. So, and like I said before, the sigma factor must bind RNA polymerase before contacting the promoter. I think that's all I really, wait, I have my notes on my phone. Oh yeah, okay. Other sigma factors expressed and used for specialized are expressed and used in specialized situations like a stress response. So that's why they're pretty general, because the organism will usually need them for a lot of different responses in the organism body or whatever it is, E. coli, bacteria, cell, whatever. But yeah, that's basically sigma. So sigma 70, to get a little more specific. like I said, it's necessary for the initiation of transcription in bacteria. So it's a protein that binds RNA, that allows RNA polymerase to bind to the promoter. So basically, it binds RNA polymerase, and then it takes RNA polymerase to the promoter to allow it to bind there. So RNA polymerase will be able to do that without sigma 70. It is sequence specific, so remember it's not gene specific. It can bind to a lot of different promoters, but it is sequence specific, which means that it binds at these two boxes called the -35 box and the -10 box. They're within the promoter. You can see them on this and the diagram, those highlighted like green nucleotides. That is the two sequences that are within the promoter that sigma 70 is looking for when it binds to. So the -10 box and -10 and -35 just relate to the position of the nucleotides within the DNA strand. So in relation to the start site. So if you look in the bottom diagram, that +1 would be where the start site is. And then -10 and -35 are just upstream of that. So that's why it's negative to the left of it. Anyways, so it binds to these two boxes. The -10 box is the Tata box, and initial unwinding takes place at the -10 box because there are more A and T base pairs. So I don't know if you guys remember from like Bio285 or something, but adenine and thymine only have 2 hydrogen bonds connecting them, whereas guanine and cytosine have 3, which means that A and T base pairs are easier to break, whereas G and C are harder because they have more bonds. So sigma is sequence specific. It binds to these two boxes within the promoter. That's what it's looking for. And it does this to regulate a lot of genes, and it can bind to various sequences. So that's why the affinity changes depending on the sequence, because it depends on what the gene needs sigma 70 for. And that's just the way that it regulates binding and initiation of transcription. Do you have any questions so far, Dov?
So sigma 70 is a general transcription factor, correct?
Correct.
Wasn't it stated that general transcription factors can bind to like many different promoters?
Yes, but it looks for the sequences within the promoters.
Okay,
So
it
yeah,
can
this,
bind,
sorry, go ahead.
sorry, I was just so it can bind to various different promoters, but those promoters need to have the Tata box at -10 and the other box at -35.
Right, exactly.
Okay, great.
Awesome. Okay, so sorry, gene regulation in prokaryotes. So they use what's called an operon. I know when I took 151, we talked about the lac operon, but in this class we're going to talk about the trip operon. So operons are basically just a set of genes transcribed under the control of a single promoter. So like I mentioned before, general transcription factors can bind to multiple promoters, but in operons it's under the control of one promoter. So this one promoter controls the operon. The genes are transcribed together. Usually they function in like the same process, so it's usually a group of genes that do like similar things. Also, just the term polycistronic mRNA is a single mRNA that codes for multiple proteins. So this is a big difference between prokaryotes and eukaryotes because we do not have polycistronic mRNA. We only have monocistronic mRNA, which means that our mRNA can only code for one protein, whereas in prokaryotes, their RNA can code for multiple. And then this diagram kind of shows you have the promoter, which is that P region, the operator is after the promoter. And that's, so the P plus the O is the control region of the operon. And the structural genes, you have ABC, they usually do something similar together. Transcription will then occur and the mRNA will be able to translate into 3 different proteins, protein A, B, and C, based on the operon. So the transcription of operons is controlled by different transcription factors. You have activators or repressors. So activators increase transcription rates and repressors prevent transcription from occurring. And they can be turned on or off depending on what certain molecule they are bound to, which is we will talk about with the trip operons. This will make a little more sense when we use the example of trip. You can go to the next slide. So just a little bit more before we talk about trip operon. Each operon contains regulatory DNA sequences that act as binding sites for regulatory proteins. So the promoter is where the RNA polymerase binds, like we talked about, and the operator, which is different, is where repressors bind. So repressors, like I mentioned, will stop transcription, whereas activators will bind to the DNA somewhere before the promoter is like situated. So if you look at where Sebastian's cursor is, the activator is that would bind before the promoter. So it's that green blob. So it would bind basically before RNA polymerase. So once it's activated, the whole thing will just go through the entire, like it'll just go through the whole transcription because the activator told it at the very start. Whereas the repressor will bind to the operator, which is after the promoter or after where the RNA polymerase binds. So it's kind of like a stop sign. So the RNA polymerase is like, oh, okay, can't do transcription. So that's the main difference between where they bind. So just try to remember that the activator is before the promoter and the repressor would bind after the promoter. And then as you can see on the diagram, no transcription, there's no mRNA made. No proteins will be made. So operons can be inducible or repressible. So inducible means that the operon is usually off in its normal conditions, but it can be turned on in the presence of a specific molecule. So I don't know if you guys remember, but the lac operon is inducible, which means that it's usually off, but in the presence of lactose, it'll be turned on. Whereas the trip operon is repressible. So repressible means that the operon is usually on in its normal conditions, but in the presence of a specific molecule, in this case tryptophan, it will be turned off. So we're going to focus on the trip operon. It's repressible, so it's usually on, and it'll be turned off when tryptophan is in like the surrounding area. Do you have any questions so far, Zev?
So is this a form then of like prokaryotic transcription regulation, where like in eukaryotes they're like the histones, but
Yes.
I guess in
Yeah.
prokaryotes would be
This is
the
basically like the whether or not transcription will occur, basically. Yeah.
Okay, great. Thanks.
Okay. So the trip operon, so just a little key, trip means tryptophan. Tryptophan is an amino acid, it's necessary for protein synthesis. So like I said, in the presence of tryptophan, it will turn the operon off. So, but there's different scenarios, and this is kind of where it gets a little bit confusing. So just the short summary is that low tryptophan levels means that the bacteria don't have access to tryptophan, so they need to make proteins through transcription, translation, the whole, central dogma process to make more tryptophan. So this means that the trip genes are on, the trip operon will go through with transcription. When there are high levels of tryptophan, there's a lot of tryptophan around. So the bacteria will be like, okay, I don't need any more, I have plenty. So they don't want to, they wouldn't want to waste energy, which is like the next question I ask, by making more tryptophan when they have plenty. So the trip operon will be off. So why does control matter? Like I just said, if you have so much tryptophan, you don't want to be wasting more energy or resources than you have enough already. That's why high levels will turn it off. And then I think we're going to the situations and stuff. Oh, okay, then the structure, then the situation. Okay. So yeah, here's just the structure. It's just a little bit bigger from what we talked about. You have the promoter and the operator. That's where the RNA polymerase binds to the promoter, and the repressor binds to the operator. So this is just the whole thing. There's a section after the operator which is the attenuator. This is a very simplified version. We're going to go a little bit into it in the next few slides. But then these TRPE, D, C, those are all the genes that code make protein for tryptophan. So this DNA will turn into mRNA that will then be able to make the genes for trip A, B, or the proteins for A, B, C, D, E, whatever. So if you look here in the bottom left, the R, so this is the repressor. So when there's no tryptophan, there's no repression because there's nothing binding. So there's nothing that will bind to the repressor to tell it has enough. So this red triangle is the tryptophan molecule in this certain diagram. So if there isn't enough tryptophan around, it's less likely tryptophan's going to bind to the repressor. The repressor won't bind to the operator, and transcription will occur. Whereas if there's a lot of tryptophan around, so say this page is covered in red triangles, it's more likely the repressor will bind to the tryptophan, and as a result, it will bind to the operator and be like, you need to stop making. Does that make sense so far? Do you have any questions? Let me know.
Yeah.
Okay, cool. Awesome. So, okay, situation 1. So like I said before, high levels of tryptophan mean that the repressor will bind to the operator and will stop transcription. This is I think the easiest, in my opinion, the easiest scenario. When you have really high levels, you don't need any more. It binds to the repressor is like, okay, we can stop. It binds to the operator, RNA polymerase can't start transcription. None of these proteins for tryptophan will be made. And that's basically that. That one's, I think, for me, is the easiest one. Also, if you hear Laura ever mention a co-repressor, she's just referring to tryptophan because that's what's binding to the repressor itself. So it's just a co-repressor. So, okay, so like I mentioned the attenuator before. So the attenuator region of the mRNA is, it basically forms these like hair loop or these stem loops. And it's important for maintaining resources. So if attenuation occurs, that means that there's an early termination of transcription. So this kind of is when it gets tricky, when there's like a medium amount of tryptophan in the cell, because it's like either there's still not enough for the cell, or it's like there's too much. But it's like that medium region when it's like not like black or white, it's like gray area. This is when kind of attenuation occurs. In a scenario in which attenuation would occur would be something like you started out with low tryptophan, so you started transcription, and then you got plenty, and then you halt it. Like after RNA polymerase already started transcription, attenuation would cause like the stop of transcription. So why do prokaryotes use attenuation? It's because if you remember, they transcribe and translate proteins at the same time. So mRNA is being transcribed at the same time that it's making proteins. So while mRNA is being transcribed, it can also be making the tryptophan proteins, and then like halfway through, it could be like, all right, we have plenty, we need to stop transcribing this mRNA. We don't need it anymore. So just some quick terminology. This, if you look on this diagram, it's a little confusing at first when you first glance at it, but just look at the numbers. So you have this 1, 2, 3, and 4. So 1 is that blue region, 2 is pink, 3 is purple, and 4 is yellow. Also, Sebastian, it's 6:15. Do you have to go? Or are you good?
Sorry, could you repeat that?
I said, do you, like, if you need to go at any time, you're totally fine.
Oh, okay. I think I'm okay for now, so I could stick around.
Okay, yeah, all right. Just let me know.
Sure.
Like, you can also just like jump in and interrupt. You're totally fine. So yeah, so look at the numbers. You have 1 blue, 2 pink, 3 purple, 4 yellow. Regions 1 and 2 are complementary. I can never say that word. 2 and 3 are complementary, and 3 and 4 are complementary. So basically, attenuation is a form of regulation that determines how long the mRNA transcript will be. This is where it gets a little hard to explain. But basically, if you have this 1 and 2 binding together in the first scenario, transcription will be paused. If 2 and 3 bind together, like in the second scenario, then that means transcription will continue, which will lead to translation, which will lead to more tryptophan proteins being made. And then 3 and 4 would result in that early termination of transcription. So this isn't just mostly important for saving resources. It's kind of an evolutionary thing that prokaryotes kind of have over us, because we just transcribe in the nucleus and then we translate in the cytosol. We have to move our mRNA transcript out of the nucleus, whereas prokaryotes do it at the same time. So that's why they have this type of regulation to kind of monitor when it needs to stop, when levels of tryptophan are getting too high, when they start out low. There's a little bit more of a recap at the end, which will kind of tie that together, but I know attenuation can be a little confusing at first, so if you have any questions, just let me know. It's really fine. It's just these three scenarios where the transcript or the DNA kind of just aligns in this hairpin to give like a signal to the cell whether to pause, continue, or terminate. That's basically what attenuation is.
So the degree to which like there's amounts of tryptophan will determine which of these regions fold onto each other.
Yes.
And does it go in order? Like do 3 and 4 fold first, and then 2 and 3, then 1 and 2?
It depends, honestly, on the scenario. So if mRNA is currently making too much tryptophan, then 3 and 4 will go together. And then I don't know, I personally don't know when, Sebastian, you might know, when 1 and 2 would occur, when it would need to pause. Because I don't know what would lead to it to pause, but I know 2 and 3 would occur if there's not enough tryptophan. You need more transcription translation. 3 and 4 is when there's too much. I don't know, Sebastian, do you know about 1 and 2?
Yeah, so regions 1 and 2, if regions 1 and 2 bind together, that means the regions 3 and 4 will also bind together. And
Oh, you're right.
yeah,
Forgot about that. Yep.
yeah, if you get a situation where 1 and 2 bind together, 3 and 4 are like evidently going to bind to each other also. And if 3 and 4 bind to each other, that's how it results in a termination like this. But if 2 and 3 bind together, if 2 and 3 ever bind, um
Then 3 and 4
one
can't bind, and 1 and 2
exactly.
can't bind, yeah.
Exactly. Because 2 and 3 are binding, 1 can't bind to 2 because it's already paired up with 3, and 4 can't bind with 3 because it's already paired up with 2.
So is this attenuation then because is it then determined by like the rate at which tryptophan binds to the attenuator region? Like how does it, because I would think like if it binds, then it would shut the stuff off, but it obviously doesn't work like that because there's like a gray area. So I'm wondering what that mechanism, if you have any idea.
I think that's actually on the next slide.
Yeah.
Cool.
Yeah, so we can go into that. Okay, perfect. So situation 2, this is the low levels of tryptophan. There's 2 things that can kind of happen, is either there's too much made or not enough. So basically the repressor is not bound to tryptophan, so it won't bind to the operator, so transcription will begin. So if no attenuation occurs, which means that 3 and 4 together, or that hairpin, then you get a full transcription and translation of all the mRNA, all the genes that code for the proteins will be made into proteins, and you'll get more. So this here shows 1 and 2, which is that pausing. So the RNA polymerase, I think it makes it to here, right? At the group, like this, yeah, it makes it to there, right, Sebastian? And then it makes like that hit, it makes the hairpin and gets stuck. It's like the frowny face.
Yeah.
May made the slide, so I'm not really sure. I'm trying to like.
Yeah, so in low level, okay, so this yellow thing is your RNA polymerase. It's just transcribing whatever. Because in prokaryotic cells, transcription and translation happen at the same time, this ribosome, it's kind of just following along. It's directly behind the RNA polymerase. It's right behind it, just following it, translating as this guy is transcribing. In this situation, you have low levels of tryptophan. So what's going to happen is at this UGG, these are 2 codons, the code for tryptophan. And again, this is a situation where there's a low level of tryptophan. So if there's a low level of tRNA tryptophan, the ribosome is going to get here and it's going to be like, damn, it's stuck because it doesn't have enough tRNA tryptophan. If it's stuck here on region, actually, I used to use a different diagram, but I think it's region 2.
Yeah.
If it's stuck here on region 2, that means that it cannot bind with 3. So what's going to happen is that region 1 and 2 are going to bind, because the reason 2 cannot bind with 3 is because
The ribosome gets stuck on 2, doesn't it?
yeah, it gets, wait a minute. Yeah, it gets stuck on 2. Sorry, I usually use a different diagram, so
No, you're good.
I'm
You're
trying to figure
good.
this out. If 2 gets stuck on 3, or I'm sorry, if the ribosome gets stuck on 2, then 2 actually will base pair with 3, and it'll be this scenario, which it'll pause for a moment, but then once the cell does eventually find enough tRNA tryptophan, it's going to continue. So the problem is if it gets stuck on region, if it gets stuck on region 3, I believe, that's where you're going to have attenuation. But in this situation, if it gets stuck right here around region 2, it's going to pause, but you're going to end up with the full mRNA transcript, if that makes sense.
Yeah.
So,
Thank you. I just
yeah, no worries.
attenuation's always, it's not my favorite. But
I'm
do you
like
have
the
any-
dumbest person ever. So I studied this like so hard and I like engraved this into my, I will go into my grave knowing how attenuation works, only because I studied this so much because I didn't get it.
Yeah, it's a lot.
Yeah,
It wasn't on the MCAT, so it was something I had to learn that was new.
that's a relief.
Yeah. Okay. So yeah, so basically that's that one scenario. I'm just
Yeah.
the hairpin. Okay. I'm just trying to think. When we get the full transcript,
Yes. That's when you have low levels of tryptophan.
yeah,
Very low
because
levels.
you have 2 and 3 bound together. Okay, yeah, we just described all of that. With when, because this little gray or bluish thingy is the
Oh,
ribosome that's making the proteins off of the transcript.
yes. Sorry, I just realized the ribosome, this thing,
Mhm,
it's stuck on region 1, hence why I
mhm,
like the colored one. So when the ribosome gets stuck on region 1, that allows for 2 and 3 to base pair, which is what you
right.
see here. And now then 2 and 3 have base paired, you have a continuation of transcription and translation.
And you get the full mRNA transcript. You
Exactly.
get
The
full,
problem, right, the problem which Rachel will describe soon is if the ribosome stops here on region 2, that's where the
right,
problem comes in, which Rachel will talk about.
right. And then wait, go back to the first one real quick.
Sorry.
In the bottom left, so this just shows that pause that we're talking about. I'm sorry, bottom, no, yeah, bottom left. I don't know my lefts and rights. So when that 1 and 2 form, when you get that pause, it allows the ribosome to just catch up with the RNA polymerase, because remember they're moving to the left here. So that's why it gets stuck. So it just allows that quick pause. That's what this first scenario is showing. And then the second scenario that we just talked about is the ribosome is getting stuck on the 1 region, allowing 2 and 3 to base pair. And so when they pair, 3 and 4 can't pair, which means that transcription will continue all the way through, and we'll get the full mRNA transcript. And remember, this is all with low levels of tryptophan. So this is when there's not enough, you need more. That's why you want the full mRNA transcript. Okay, now we can move on to the next slide. Okay, so this is the weird gray area where you start with initially low levels and then you end up with high levels of tryptophan in the cell. So attenuation, always remember that attenuation would occur after transcription has already been initiated. So it's not like when we talked about the repressor binding to the operator, that stops transcription from ever starting, whereas attenuation stops transcription after it has already started. So like we said before, the 1 and 2 hairpin form, the ribosome, which causes this pause, allows the ribosome to catch up to the RNA polymerase, because they're transcribing, translating at the same time. But so you have to remember in this case, transcription started because there were not enough tryptophan in the cell to tell it to stop and not do transcription. But by the time the ribosome catches up to the trip codons, there's now enough tryptophan to make that to be made, so the ribosome won't be stuck on region 1 like in the last scenario when there wasn't enough. But now there is enough. So what happens is the ribosome gets stuck on the region 2, or that stop codon, that UGA in this, where Sebastian's cursor is. So now it's stuck on region 2. So when region 2 is blocked, it can't bind to 1 and it can't bind to 3. So 3 and 4 can hairpin and make this termination signal, which stops transcription and results in this short mRNA transcript right there. You can see it's like now it's detached from the RNA polymerase. But when 3 and 4 are allowed to hairpin, the RNA polymerase will dissociate from the DNA and the mRNA will stop translating, and you'll just have this short mRNA transcript. So this is that case where it started, if in order for transcription to even start, the levels of tryptophan had to have been low. But once you get enough as you're translating, because they occur at the same time, this 3 and 4 hairpin will form and termination of transcription will occur. Do you have any questions, Seth, at all? I know this is kind of a lot.
Yeah, I'm not totally getting it, but I mean, I'll watch the lectures and actually do the assignments, and that'll probably help.
Yeah, Laura might explain it a little better, or even coming back to watch this section of the review, like after you
Yeah.
watch
This is arguably like one of the most confusing parts of this class, the attenuation sequence. And again, like you're always welcome to come to our office hours, and we could probably like continue explaining it. Yeah, it's all just very like contingent on concentrations.
Yeah, definitely. The recap in the next slide will help a little bit. So let's do that. So these are your 3 situations. So you'll start with, so like I said, the high levels of tryptophan I think are the easiest because the repressor binds to the operator when it's bound to tryptophan because there's a lot. So there's just no transcription occurs, no translation occurs, just a big stop sign when there are lots of tryptophan molecules around. Situation 2 is that low level. Transcription will occur because there's not enough tryptophan to bind to the repressor. So the repressor will not bind to the operator and will not be that stop sign. So the cell will get the go to do transcription. Because the ribosome will pause on that 1 and 2, or that one section, 1 and 2 will form a hairpin just to allow it to catch up. And then as it's making tryptophan, there's still not enough. So 2 and 3 will be able to hairpin, and you'll get a full transcript of mRNA and full translation of the tryptophan proteins. And then situation 3, in my opinion, is the most kind of confusing because it's that little gray area. So you start with a low enough concentration of tryptophan that transcription does occur, but as you're going through transcription and translation, the cell is like, okay, now we have enough, we don't need any more. It forms that 3 and 4 hairpin to signal to stop. So you stop transcription, translation, you make a shorter transcript. Does that kind of make a little bit more sense when you see the 3 scenarios together?
So the hairpins are good? Because I thought for some reason that when those formed, it was like a bad thing.
It depends on the scenario. It's not in terms of like good or bad. It's just how much tryptophan does the cell need. So if you have that 2 and 3, you don't have enough. If you have 3 and 4, you have too much. Does that help a little bit? It's just, I just like to see them as kind of like a signal really for the cell, like which signal it's giving. 2 and 3 means go, 3 and 4 means stop.
Yeah, kind of. I'm little by little.
Yeah, no worries.
Yeah, it'll make sense. It took me a long time to get it too, so don't worry. And then I think oh yeah, just any questions? Oh no, practice problems. Okay. So if region 4 is deleted, what do you think the size of the mRNA transcript will be? And like, obviously, if you don't know, totally fine, don't worry. But what do you think?
The size region. It would be fully trans.
Yeah, because you wouldn't be able, regardless of whether it pauses or not, there's no termination sequence to occur. So 2 and 3 can bind together and make that continued transcription translation, but 3 can't bind in anything else. So there would never be an early termination. So I would expect you would get a full mRNA. Okay. If the UGG sequence is mutated, there's no. I don't like this question.
I actually wrote these questions.
Oh my God.
I'm sorry. These are
No, Sebastian, I love your questions. I thought you wrote this question.
No,
I feel like I remember this from like one of the reviews from last semester.
Yeah, no, you're
Do you
totally
want to
right.
go over this one? Because I don't know it.
Yeah, it's essentially the same concept as if so let's say for instance your UGG sequence, that's the
That's one, right? Yeah.
yeah, that's the tryptophan. So like Rachel mentioned, if your polymerase is transcribing and then it realizes it has enough tryptophan to where it can like very cleanly, very easily translate straight through this UGG region, then the ribosome is going to stop at the UGA, at the stop codon. If it stops at the stop codon, then that's how you're going to get this attenuation thing. So the question is just saying, if you mutate this region that codes for like 2 tryptophan anticodons, if you change them to where it now requires 2 cysteine anticodons, and the problem says you have plenty of, wait, did I say that UGG? And there's no, oh, sorry. So you mutated them to cysteine anticodons, and there's no cysteine anticodons around. What do you think the size of the transcript will be?
I have no idea.
I thought you left the session for a second. Sorry.
I'm
No,
not,
you're fine.
so what does the cysteine do? I thought, so do we want tryptophan or not?
Yeah,
Like, I thought it's like for maximum transcription, we don't want tryptophan.
right. This maybe this was just like a bad way of phrasing it, but essentially, if you mutate the tryptophan region to where instead of requiring tryptophan, you actually need cysteine, and you have no cysteine around, what's going to happen is you're going to produce the full length transcript. And the reason for that is because it's going, the ribosome is going to be translating. It's going to get to this is the tryptophan region, but in the problem, we mutated it to cysteine. The ribosome is going to get to the cysteine region. It's going to be like, damn, I don't have any cysteine, and it's going to be stuck there. And because it's stuck on region 1, regions 2 and 3 are then able to come together to hairpin. And if 2 and 3 come together, then you will end up with the full level of transcript, or I'm sorry, the full mRNA transcript. It's only regions 3 and 4 hairpin loop that you will get a short transcript, that you will get early termination. So in this question, if you mutate the tryptophan to cysteine, it's essentially the same deal as if you don't have a lot of tryptophan around. It's exactly the same deal, actually. If you don't have enough tryptophan, the ribosome will get stuck here because it doesn't have enough tryptophan. That will result in 2 and 3 pinning together, and if 2 and 3 pin together, you get the full length transcript. Does that make sense?
Yeah.
Word. Okay, I won't use this question again for future reviews. Because
it makes sense when you explain it. I just honestly forgot how to explain it and not sound like a bumbling idiot.
No, that's totally fine. I know you hate it, so I won't use it anymore.
You're just too smart, Sebastian. It's fine. Okay.
I don't think so.
And number 3, this is the last question, Seven, and you're free to go unless you have other questions. Totally fine. But number 3, true or false? Attenuation will only occur when TRP levels are high. What do you think?
Well, they have to be high at some point. So true?
So this is that trick question. So attenuation. So if tryptophan levels are high, I guess if you want to specify,
it can start low, but it would has
right. But if I guess in this question, maybe we should clarify that like TRP levels are high like from the start. But if they're high from the start, attenuation will never occur because transcription won't occur. But you're right. If it starts low and then high, attenuation will occur when TRP levels go from low to high.
All right. So, yeah. So attenuation will not occur if they're high because then just nothing.
Transcription would never occur
the
in
repressor
first place.
is bound.
Right.
So yeah,
Okay.
this was actually a question out of Laura's quizzes. I did not write this one, but
No,
I
I know
thought
you didn't. It was from Laura's quizzes, but yeah, it's one of those trick questions where she's expecting you to think if by just saying when trip levels are high, you should just think, okay, transcription won't occur, then attenuation won't occur, because attenuation happens after transcription is initiated. So that's the main takeaway from that. And then if you have any other questions, obviously you can shout them out, whatever.
Don't think so.
Okay, cool.
But-
And then we just have our info and our fun pictures. But yeah, so you can always also email us or come to our help sessions, or Laura's help sessions too. But yeah, that's all we have tonight.
Great, thanks. Do like people usually come?
We've had one person per session. So we've had one person went to the first one person went to the second one, and then the same thing. One person came Wednesday, one person came Friday. So
Well, this is
it's-
great.
So
Yeah.
we have an
good
average of
time.
one person per review.
yeah.
No,
So,
it's good to be able to like ask questions during the
yeah, I think
lecture.
it definitely helps. I think more people come to our exam reviews, but we'll see.
Oh geez, isn't there an exam like
On
this
the
week?
27th,
Oh,
Christmas. No, I'm kidding. Yeah.
okay.
Well,
That's
thanks.
all we have.